Hello student, today class is nucleic acid. So what is nucleic acid? It is a macromolecules and these macromolecules consist nucleotides. So nucleotides is the unit of nucleic acid and the nucleic acid consists the many nucleotides and nitrogenous base, sugar and phosphate is collectively called nucleotides and nitrogenous base plus sugar is called nucleoside. That means nucleotide means nucleoside plus phosphate and nucleoside means nucleotide minus phosphate and sugar and phosphate is the backbone of nucleic acid. This nucleic acid is two types, deoxyribose nucleic acid that is DNA and ribose nucleic acid that is RNA. So DNA and RNA, this is also a genetic material, such as in virus RNA is a genetic material and in human being or other animal DNA is a genetic material. So DNA RNA is a genetic material and difference is that DNA consists deoxyribose nucleic acid and the sugar is deoxyribose and RNA consists ribose sugar. So this is the difference. DNA is generally double helix, RNA is generally single helix and DNA follows the Chargaff's rule. RNA not follows the Chargaff's rule. DNA is Fulgen positive, Fulgen is a di, and RNA is a Fulgen negative. So this is the difference between DNA and RNA. And most important difference is the DNA consists adenine, guanine, cytosine, and thymine as a nitrogenous base. But RNA consists adenine, guanine, cytosine, and uracil at the place of thymine. So there are many differences between DNA and RNA. DNA consists nucleotide and nucleotide is nitrogenous base, sugar and phosphate. And these nitrogenous base consists two types. One is purine which is double ring and one is pyrimidine that is single ring. The purine is the adenine and guanine and it consists the main ring hexane and another is pentane that is imidazole ring. And it consists the 9-member ring. Out of 9, 5 is carbon and 4 nitrogen. But pyrimidine consists cytosine, thymine, uracil and it is the 6-member ring. And out of 6, 4 is carbon and 2 is nitrogen. And the next sugar attached with the in purine N9 and C1. N9 means N9 of the adenine or guanine that is purine and C1 of the deoxyribose sugar in DNA and ribose sugar in RNA. But N1C1 bond that is glycosidic bond present in between pyrimidine and sugar. So purine and sugar that is the N9C1 glycosidic bond and in between pyrimidine and sugar N1C1 glycosidic bond is present. So these are the nucleoside because nitrogenous base attached with sugar that is nucleoside. So nucleoside means nitrogenous base plus sugar. And sugar and phosphate is a backbone. These are the backbone. And according to this, these forms the nucleotides such as ATP, GTP, CTP, TTP, UTP. These are the nucleotides. ATP is also called adenylic acid, GTP, CTP. So adenine combined with sugar to form adenosine and adenosine combined with 1 phosphate that is AMP, 2 phosphate that is ADP, 3 phosphate that is ATP. So these are nucleotides. Such as guanine. Guanine combined with the sugar that is guanosine. And this guanosine combined with 1 phosphate that is GMP that is nucleotide. And such as CTP, TTP and UTP. These are the also nucleotide. So adenine combined with sugar, adenosine. Guanine combined with sugar, guanosine. Cytosine combined with sugar, cytidine. Thymine combined with sugar, thymidine. Uracil combined with sugar, that is uridine. And these uridine combined with phosphate, that is uridylic acid. Such as adenosine combined with phosphate, that is called adenylic acid. So these are the nucleotides. And many nucleotides to form nucleic acid, because nucleic acid is a polymer of nucleotide, and it forms different type of DNA. According to Watson and Crick model, the DNA is a helical, but in case of bacteria, DNA is a circular and double-stranded, and it is a linear DNA. That is, helical DNA is a linear DNA. And different type of DNA. Most common DNA in human body or animal body is bDNA, that is balanced DNA, which is anti-parallel, because 5 primers start to end 3 primer and 3 primers into 5 primer. So these are the anti-parallel DNA. Balanced DNA is anti-parallel DNA. And 10 base pair consists one pitch or one path, and it 10 base pair consists total distance is 34 angstrom. And in 34 angstrom, 10 base pair is present. So between base pair distance is 3.4 angstrom. And 10 base pair total degree is 360 degree, because one pitch. So 36 degree angle present between the base pair. So these are the data. That is the balanced DNA. cDNA, that is complementary DNA, present in the retrovirus. pDNA, that is palindromic DNA. So most common DNA is a bDNA, according to Watson and Crick, and it follows the Chargaff's rule. Chargaff's means adenine is equal to thymine, because adenine combined with thymine, and C is equal to G, and total purine, that is adenine plus guanine, is equal to total pyrimidine, cytosine plus thymine, and A plus T is not equal to C plus G. So these are the 4 rules of Chargaff's. So these are called Chargaff's rule. And the bonds present in DNA is mainly 3. First is hydrogen bond between nitrogenous base, such as A combined with T, that is the double hydrogen bond, and C combined with G, that is triple hydrogen bond. So first is hydrogen bond between nitrogenous bases. Second is glycosidic bond. Glycosidic bond present between the purine or pyrimidine with sugar, such as N9C1 glycosidic bond in purine and N1C1 glycosidic bond in pyrimidine. And third bond and most important bond is phosphodiester bond. This phosphodiester bond present between the nucleotides, because one phosphate combined with the sugar or sugar combined with the phosphate. So these are the phosphodiester bonds. So 3 type of bond present in DNA. And the DNA most important property is called TM value. More GC, more TM, and less GC, less TM. Because GC means guanine cytosine, more hydrogen bond, and AT, this less hydrogen bond. So more GC, more TM, less GC, less TM. That is called melting temperature. And denaturation property of DNA. That is, if DNA has high temperature, then denaturation takes place. So denaturation property is also a most important property of DNA, that is denaturation. And it absorbs the ultraviolet ray. It absorbs the DNA, absorbs the UV ray also. So this is about the overall nucleic acid and nucleotides. And other is RNA is also a nucleic acid. That is, the difference is ribose sugar, and it consists messenger RNA, transfer RNA, ribosomal RNA. And the most common RNA is messenger RNA. And the RNA which involved in protein synthesis is ribosomal RNA and tRNA. tRNA is also called soluble RNA or adapter RNA. And RNA is a genetic material given by Frankel and Condrat in the tobacco mosaic virus. So some virus has a RNA is a genetic material, a DNA is absent. But where DNA is a genetic material, RNA is non-genetic, such as messenger RNA, transfer RNA, ribosomal RNA, a small cytoplasmic RNA, small hnRNA, heterogeneous nuclear RNA. These are the different type of RNA. So DNA and RNA is a genetic material and it is nucleic acid. Thank you.